Good morning. Welcome to the New York Psychoanalytic Institute, Arnold Pfeffer Center for Neuropsychoanalysis. I have to make one announcement before we begin. On the instruction of Maggie Zellner, I must remind you that our 12th Neuropsychoanalysis Congress will be held in Berlin on 23rd to 26th June. The title of the Congress is Minding the Body. It'll be opened by Wolf Singer, and among the other main speakers are Bud Craig, Antonio Damasio, Peter Fonaghi, Vittorio Galesi, Marianne Leutzinger-Böhleber, Jaak Panksepp, Mark Solms, Patrick Willemer, and others, und anderen. Okay? You all have to come to that. But today, you've come to something else. You've come to Jaak Panksepp's presentation. The title is Affective Continuity, ? From Seeking to Play, Science, Therapeutics, and Beyond. Jaak Panksepp truly, that old cliche really does apply. He needs no introduction. He's super duper famous for very good reasons. But I will just mention probably his main academic position. He's the Bailey Endowed Chair of Animal Well-Being Science at the College of Veterinary Medicine at Washington State University. He's an absolute genius, and we're very lucky to have him here. And here he comes.
Thank you very much, Mark, and thank you for coming out this morning. And let me have a promotional here. This is a long book in the making, a book that's taken a long time in the making, and Norton has it now, which is a slight updating on affective neuroscience, along with Lucy Biven, who's many of you may know is the daughter of Charles Brenner. Yes. She's in England, works with the National Health Service as a psychotherapist. So she was supposed to add the therapeutic impact of affective neuroscience, but I don't think that quite happened for various reasons. But that's a different story. The story I would like to share with you this morning is a Darwinian story about the layers of the mind and how we might keep the layers of the mind and brain organized in a coherent way. And here's my spin on it. There's a primitive mind in all of us, and a more modern mind, and a very modern neocortex in evolutionary time. And we are communicating via the neocortex, but we are still living in the ancient recesses of our minds when we are alone by ourselves. And the evidence is quite overwhelming that our passions and our intense feelings, emotions arise from the most ancient layers of the brain. And that, for some reason, has had a very difficult time taking hold in the modern zeitgeist for various reasons, including the heavy weight of behaviorism that's still with us, and the light weight of cognitivism, which is overwhelming us. And they're both very important approaches, but not quite as important as what Charles Darwin gave us, the first modern book on emotions. And I'm sure most of you have read it, and I'm sure he would have done some brain research too if the time had been ripe. So I'm going to try to cover 5 main issues. A behavioral perspective on these rewards and punishments that the behaviorists use to train animals and humans. And that started with Thorndike's law of effect, which was originally the law of affect. But the affect was taken out of the law, and only a causal effect was left behind. And then there's this wonderful probably most sophisticated and most complex emotion of them all, that some people still call the brain reward system, which leads to many paradoxes that people often don't face up to. It is a system that's manifested at all layers of the mind, and usually the discussion right now proceeds at the highest level, which I will call the tertiary process level. But at the primary process level, it generates a certain kind of feeling that is present whenever we desire something, whenever we look for something. It doesn't matter what it is. It's a very general purpose system to obtain the resources of the world, whether they're nuts or knowledge. And it would be wonderful to have a direct measure of this system in an experimental animal so they can tell us where they are with respect to seeking space. And I think we have discovered a measure, which is a 50 kilohertz ultrasonic vocalization that we also call rat laughter, and probably is evolutionarily related to infantile human laughter. There's every indication of that, which is a bit hard for many people to swallow, but that's where the data is pointing us. And I'm a very data-oriented guy. This system also participates in all our social processes. We have various social aspirations, from lust to the products of lust, babies that need care to survive. And this system is engaged in taking care of babies. And it is at the very heart of play impulses. So that's why I have that question mark, the ambiguity of this emotional process. And finally, I think understanding of this system should have larger consequences for psychotherapeutics. And I should not be standing up here suggesting what they are, but I have no other option. Lucy Biven was supposed to do it for the book, but I ended up having to do it for the book. So the first theme, just a little history that you all know, the law of effect. What is that? Well, first of all, just general principles. The fundamental, the primary process affective systems of the mind do reflect various evolutionarily adaptive functions of the brain. They were useful for survival, and if they came first, everything else is still dependent upon them. And I think philosophy is paying attention to that possibility. I think cognitive science is beginning to realize that what came first guides what came second. And I think our legal systems also have to deal with it in the sense of intentionality, res mensa, and it's not just the fact of a criminal act, but it's the intention behind it that produces all the confusion. So in our modern search to understand the mechanisms of the brain and mind, we neuroscientists often choose to forget that widespread neural networks generate various psychological processes that regulate behavior. So there is something in the organization, large-scale organization, that the neuron doctrine does not get at. That I think everyone is beginning to believe that we need a network doctrine, but we do not have the sophisticated measures for network dynamics the way we have for neurophysiological dynamics or neuronal dynamics, action potentials. And that's a bit of a tragedy. And if one were to ask, what is the very best measure of a network activity in the brain that is within the interesting emotional motivational domain, I think one has to conclude it is instinctual emotional behavior. It is so characteristic in species and across species that you easily recognize it, and it's built into the nervous system, and it was seen as an output, like a knee-jerk reflex. But the mountain of evidence, and it's really a mountain of evidence, indicates that the instinctual apparatus that generates these behaviors also generates the affective feeling. Now that's a very a very efficient design characteristic. Shouldn't eat a bun right before. Little fake flakes get in your throat and tickle it there, and then you try to suppress the feeling. Much better to take a drink and regulate affect. So, thinking, perceiving, feeling, and behaving, and getting depressed, which is one of our main research interests right now, are not simply properties of brain molecules and individual neurons. To understand how brains generate feelings, affect, those powers of the mind, we also need psychological perspectives. We need a network doctrine as opposed to just a neuron doctrine. So here's the law of effect by Edward Thorndike. He said, this is a quotation, of several responses made to the same situation, those which are accompanied or closely followed by satisfaction to the animal will, other things being equal, be more firmly connected to the situation, so that when it recurs, they will be more likely to recur. So if you do something and it feels good, you'll do more of it, basically. Those which are accompanied or closely followed by discomfort or annoyance to the animal will, other things being equal, have their connections to that situation weakened. You do something and it feels bad, you're going to do less of it. That's all it says. And it's an affective view. The greater the satisfaction or discomfort, magnitude of reward or punishment, the greater the strengthening or weakening of this bond, memory. And nothing more is implied by the bond at that point in time. Satisfaction was arbitrarily changed to reward, reinforcement. Discomfort was arbitrarily changed to punishment, because those terms could now be defined as objects in the world as opposed to processes in the brain. Of course, they had no access to the brain at that time, so that was a wise move. But this was originally a law of affect, and now that we have sophisticated neuroscience, we can go back to it, and we can ask, what makes a reward a reward? What makes a punishment? And the answer seems to be, it produces affective change in the nervous system. How simple can that be? But neuroscience does not accept that, because those are subjective aspects of this thing called mind that is still presumably unapproachable, because we have a desire to be control freaks. And here was the original control freak, Jaak, who was a peripheral member of the Berlin Biophysics Club. And being Jewish, he could not find a proper academic position in Europe, and he had to come to Bryn Mawr College for his first academic appointment, where he stayed there 1 year and was immediately scooped up by the University of Chicago, the hotbed of evolutionism, where there was a young student who was a psychology major, John Watson, the father of behaviorism. Watson wanted to work with Jaak Loeb. His committee said, Loeb is a little too weird. You better stick with one of the psychologists. And there, across on the eastern seaboard, was a young student who was influenced by Charles Crozier, a physiologist that was also trained in the Jaak Loeb tradition, B.F. Skinner, of course. And they gave us the most rigorous methods for studying learning, but they also chose to be radical behaviorists, that's all we had to know to have a coherent understanding, and of course that was not true. You also had to understand the brain, but they just wanted to control behavior, not understand it. They indicated that very clearly, and that's how all the other animals lost their feelings. And that's how human feelings were no longer discussed in the academy, and we are still recovering from the massive shock of that explosion of empirical data. So what do we do now with ruthless reductionism? Which was, there are things in the world, stimuli, and there are organismic responses to the world that are called behavior. And all of a sudden, neuroscience matured. The first meeting of Society of Neuroscience was in 1972. 200 abstracts. I presented one of those abstracts, and in Washington, D.C., and the jig was up. The government was pumping all its money into neuroscience and not behavioral analysis, so behaviorists became brain researchers. So we have behavioral neuroscience as an enormous field to this day where the dogma prevails. Never mind. Never. And affect is a mental representation of neural activity. And affect was right down below. So pain is primitive. One experiences pain down deep below. But our little babies, until quite recently, could be circumcised without anesthesia because they presumably did not experience the pain. Of course they experienced the pain. Now there's plenty of evidence that that trauma percolates down through the years and is not a good thing to have at a very early age. So experience has many levels. I'm sure the little baby is not aware that it's having the experience. It's only having phenomenal consciousness, having something that it is to be in the world without being able to think about it, which is awareness in a higher form of consciousness. So that was what this book was about, trying to get a clear, empirically based image of the emotional affective processes that are so important for psychiatry and psychotherapy. It was hopefully an antidote against ruthless reductionism. And reductionism is not bad. It's a very good way to approach mechanistic questions, namely how something operates. Scientists usually do not try to answer why questions, but evolution even permits that. So, the most sophisticated emotion that is built into the mammalian nervous system is play. It is built into the nervous system as an urge, rough and tumble social play, and we now know that playful behavior reflects some kind of a joyful feeling. And that meaning percolates into a lot of learning, and it percolates into maladaptive behaviors. And the most radical thing I will say today is that bringing playfulness back into the therapeutic environment intentionally with finesse can have profound therapeutic effects. That's a hypothesis. And if you can bring laughter into the therapeutic situation at critical moments, you can modify the painful memories of the past. Okay? That's what the data is beginning to show. So I'll spend a little time on these 50 kilohertz rat laughter chirps. But first of all, levels of control within the brain. At the bottom, there are primary processes. Basic primordial affects. There's subneocortical. There are various types. The ones I'll focus on and have always focused on are the emotional affects that are built into the action systems that generate emotional behaviors. They organize intentions within the actions of the organism. They are evolutionary memories. They're intentions that the organism has no choice about. It simply has to behave out those features of being. Then there's wonderful homeostatic affects. I didn't get much of a breakfast, so I thought I'd get a couple of calories to make sure I didn't collapse here. Hunger, thirst, thermal regulation, the bread and butter topics of behavioral neuroscience. And that is how I paid my dues too, until I got tenure. And then I dropped it. That's the role of tenure, to be able to do as you please. And I shifted completely to emotion research to begin mapping systems that organize social attachments, especially the pain of separation. And that's why we focused on the separation call, distress vocalization from social loss, but I will not talk about that today. Then there's sensory affects. Nice munchies here in the morning, coffee, someone giving us a neck rub or other rubs. And, the good feelings that come from the external world. And Kent Berridge at University of Michigan is doing the most wonderful work on that with gustatory responses of little rats and telling us where those feelings occur, but not trying to use the language of feelings. And he's having a bit of a tough time with that. But hopefully he'll solve it. Then on top of that, we got various secondary processes, and I'm using kind of Freudian language because it is the sweetest language for this kind of discourse. These are in the basal ganglia. They mediate classical conditioning like fear learning, which has a rich database now. And these processes are probably deeply unconscious. These processes are not. They are experienced deeply, but non-reflectively. Then, as Skinner and Watson showed us, you can generate instrumental operant conditioning systematically. So seeking of rewards has a basal ganglia, nucleus accumbens. Fear has the central amygdala. And then on top of it, you have a variety of habits. Okay? So these kinds of learning that are parsing affect into space and time, so they're not so overwhelming like in a little baby getting circumcision that fill everything. They become moments in time based on danger signals and safety signals, so you're getting affective regulation. And of course, we humans spend most of our mental world in the land of awareness, which is that we know that we are experiencing. So there's cognitive executive functions, frontal cortex, emotional ruminations and regulations more in the medial parts of the brain, and a reasonable concept of free will, namely being able to reflect on alternatives for the future based upon past memories. It is a non-problematic concept from a realistic, naturalistic, biological perspective. Philosophers, of course, want more than that, and they make a dilemma out of it. So we have these passions, and as poets have shared, and Descartes gave us that spooky image of dualism, that there's non-material mind, pure mind, mind dust. And Spinoza said, no, it's a unified material world, and mind comes from the body and the brain. And now we have cognitive neuroscience of emotions represented by Antonio Damasio, Joe Ledoux. We have behavioral neuroscience of emotions that was represented by Geoffrey Gray in England, Oxford, and Edmund Rolls, still at Oxford, at an independent institute. And affective neuroscience goes back to Paul MacLean in the most explicit form, and myself perhaps even more explicit. So what do they say about feelings themselves? Very, very different things. And most of it is in the land of opinion, not causal data. Plenty of correlates that feelings might not be relevant for a variety of situations. When you've learned and you've got habits, then you don't need feelings to guide you anymore. So the behaviorists still wish to say feelings are irrelevant. Up here, people say feelings are part of the highest parts of the brain. And who will deny that the highest parts participate and are penetrated by feelings? But the raw feelings, and there's a mountain of data for this, are subcortical built-in functions of the brain. And the law is wherever in the brain you artificially evoke a coherent emotional behavior like anger or fear, and you ask the Thorndikean law of affect question, does it mediate feelings? Rewards and punishments are the only way we can get at animal feelings. They always serve as rewards or punishments. We've known this for 40 years, but it has not penetrated the conversation for some reason. Okay? Scientists are supposed to be skeptics. That's an ultimate value. But when the mountain of evidence gets so large and you maintain skepticism, then I believe it's an ostrich-like stance. And that is still being rewarded. So here's a block diagram. We're born with unconditioned responses. That was the behaviors term. These instinctual emotional processes, the way ethologists describe them, and they are big in early development. Then we have these basal ganglia that allow feelings to be parsed into space and time. These are secondary processes of learning and memory. The mechanisms are deeply unconscious. Deeply unconscious. But the parsings are not necessarily. And on top of that, we've got the capacity to reflect and think and do all those higher mental processes. Now, this is how things look during early development, this kind of pattern. And then you grow up, and these things almost seem to disappear because we've had lots of learning and memory that has gotten larger. So we live in the land of tertiary processes, and wonderful it is. But we tend to forget the bottom because this is so big in maturity. So my friend Endo Tulving at University of Toronto, great memory researcher, has a way of looking and discussing consciousness that fits this scheme perfectly. He says there is anoetic consciousness, namely phenomenal experience without knowledge. You just have the experience, anoetic. There is noetic consciousness, probably at this level, where you develop knowledge structures, and there is autonoetic consciousness on top, which allows you time travel. We are time travelers. We have memories of the past that we can project into the future and make plans. So that is the higher level of reflective awareness. And awareness is not a good term for phenomenal consciousness. Experience is a good term. So the cognitive revolution has taken over because of the importance of this in human lives. It's just the same thing drawn differently, pyramids, in honor of Egypt. So, primary processes, big, and then the pyramid is reversed, and they seem to be tiny. But humans want their affects to be respected, and if they're not, they will have revolutions. So how are these actually organized? They're interknit like some kind of a complex sweater from the Andes with a wonderful texture. They obviously are related to each other. There's bottom-up modulation always. If you activate your emotions with brain stimulation right now, it will take over your whole mind. It will be insistent. You cannot reverse it with a willpower. It will carry you away with extreme anger or extreme fear and so forth. And then there's learning that it controls, and this learning leads to bottom-up differentiation of thoughts. And then once this develops, you've got top-down controls. So this is a non-nested hierarchy. And as your last speaker, Todd, I think highlighted, he's been pushing the vision of nested hierarchies, that what came earlier is still nested in the previous level. And these more primitive levels are nested within our higher mental processes. Nice way to look at it. Here's just the same thing, slightly differently. It gives us two-way or circular causation, doesn't force us for a bottom-up view. Bottom-up learning and development is real. Bottom-up influences on ruminations and thoughts are real. But you do have top-down cognitive regulation and top-down conditioned control. So these nested hierarchies do have to respect the primary process, the secondary process, largely upper limbic, basal ganglia, and tertiary process, largely neocortical. So, the laws of affective consciousness at the bottom, namely phenomenal affective experience, means the behavioristic, unconditioned stimuli and unconditioned responses. Sweet words as they were, they reflect primary process within brain rewards and punishments, which are affectively represented. They are value structures that are intrinsic. It's all very straightforward. How are new behaviors reinforced? As a procedure, reinforcement is no problem. You apply rewards and punishments systematically with reference to what the organism does. But reinforcers, there is no reinforcement process in the brain that anyone has ever identified. It's a word. It's a concept for something no one understands, and it could be a phlogiston, an ether, a made-up concept that has no neural reality. And I would put my money on that. What reinforcement is how affective processes influence learning and memory. And we have no research like that going on, really. Okay, that's rather surprising. So the brain mechanisms of learning and memory, secondary processes, are deeply unconscious. The tertiary mental processes, thoughts, reflections, are grounded on the previous mechanisms. So that's it for the first theme. The second theme, a general purpose seeking system. Discovered by Jim Olds and Peter Milner, totally by serendipity. They were studying the ascending reticular activating system, and their first rewarding electrode was here in the septal area, and not in the heart of the seeking system by any means. And they found that this stimulation was rewarding. And we know that the septal stimulation is a different reward than emerges from where everyone puts their electrodes now. The behavior is different, and from the human self-reports, the phenomenology is dramatically different. But the phenomenology of rats, at least, along this circuitry, is the same. Because if you have an electrode here as a cue, like a CS that predicts reward, and another electrode here, a CS minus that predicts reward isn't coming, that with little changes in tone frequency, animals pick up right away. Animals do not pick up these discriminations by stimulating two separate parts of the seeking system. That tells you psychologically it's a homogeneous process. What was it called? Well, it was called a reward, or the pleasure system, or the reinforcement system, just using traditional terms. But when you stimulate the system, the animal always goes into a search pattern. It does not look like it's being rewarded. It looks like it's looking for reward at the simplest descriptive level. So we took that seriously and called it a seeking general purpose foraging system that's needed for all resources. That concept has had a bit of a tough time. So let me just summarize the history very briefly. 1954 at McGill, these systems are discovered. And Jim Olds was pretty loosey-goosey and didn't have any behavioristic commitments, just allow the data to take him wherever it takes him, which was terrific. The first major theory, because there are already lots of paradoxes, was Tony Deutsch at San Diego. And Tony said, this actually generates two processes, kind of like a hunger drive, a need of the body, and also the satisfaction, like food when you're hungry. So it simultaneously does two things, and that explains the paradoxes. Glickman and Schiff brought us an ethological connection. And they said this is kind of a general approach system, and there's also a general avoidance system in the brain. We came along with a unified incentive hypothesis that this is the incentive function of the brain. Incentive is defined by amount of reward, the quality of reward, and the speed at which you get it. Latency to be rewarded. Want my stuff? I want it now. That's good. Don't make me wait. So there's an incentive structure for that process. And then along came the monkey wrench into the whole field by Elliott Wallenstein at University of Michigan that threw everyone into a tizzy. Because people thought now we have specific drive systems, hungers, thirsts, lusts right here, their respective rewards that of course generate these behavioral manifestations. He said it's all the same, and we were not too troubled because we already had a unified incentive hypothesis at University of Massachusetts. So the full plasticity didn't trouble us too much because of its general purpose, it'll have many, many manifestations. So I really went very strongly for that unified interpretation, and we called it an expectancy system to try to grab the attention of the cognitive community, which it did not succeed. We tried to call it foraging, which would attract the ethological community, and that did not succeed. So we said people aren't understanding what we're saying. So in the 1998 book, I decided to call it a new name, a seeking system, that now everyone uses in lowercase. But I capitalized it for a specific reason, because language is intrinsically ambiguous. People have different meanings for the most simple terms. The capitalization indicates that I am positing a real network function for an emotional process that has a variety of intrinsic attributes, and that I am not talking about the vernacular. I'm trying to generate a specialized language for the foundation of the mind. And I think most people here understand that. The need for such a nomenclature. So then all of a sudden brain dopamine was discovered, and everyone forgot the old controversies. There was a neurochemical system at the heart of this. And you should know who brought it to us. It was Urban Ungerstedt in Stockholm. And he had several papers, but this was the main one in 1971. Smack when Wallenstein, all this stuff was happening, and everyone started chasing dopamine. And after dopamine, Roy Wise, who believed there were lots of little drive systems for hungers and thirsts and the rewards, said, no, it's a unified hedonia, a pleasure concept. It's like honey on your tongue, basically. And he got burned. Rest in peace, 1993. He says there's too many paradoxes with the pleasure concept. Long story. Along came Robinson and Berridge, Kent Berridge, remember the guy that does lip smacking in rats when you shoot sugar on it. And he said, we have to distinguish wanting from liking. Wanting is the desire, liking is the pleasure that results. So even though he explicitly said, I'm not talking about anything in the mind, these are just words of convenience for communication. And I've been pursuing the seeking concept ever since 1971, and Berridge's wanting is very similar to this, but quite different, because wanting and liking are sensory concepts. Okay? This is a sensory motor concept with the motor action systems being foremost. That the organism needs to be in the world. And this being in the world is not unconscious at the primary process level. So whenever the emotion is aroused, it also provides the value of things in the brain. That's the argument, and that's what the data supports. But there's now a consensus This dopamine energized system is involved more in appetitive motivation than consummatory reward. So what is the word for the affect of appetitive motivation? It is enthusiasm. It's excitement. In its most extreme form is what the psychostimulants, cocaine and amphetamine, produce. Euphoria. That is not a pleasure concept. It's a powerful being in the world type of concept. So, but information processing, the cognitive views, took over from 1990 onward, and Wolfram Schultz, now at Cambridge, has generated this reward prediction error concept that it's hard to wrap your mind around, but let me just say, there's something to it, but it is a concept for the tertiary process. It is not a concept for the primary process. So people are talking past each other because they don't have levels of organization in mind. All these systems they're interested in are primary process systems, but they're using tertiary process language to talk about them. Okay? Wanting is a propositional type of thing. I want something, as opposed to just general wanting type of feeling, which is what the primary process does. So this is perhaps the most important emotional motivational system in the brain. First described in rats by LaCrosse Clark, 38. It's called the medial forebrain bundle. It connects midbrain, central midbrain areas with a diencephalon, especially hypothalamus, and it connects to medial frontal cortical regions, and in the rat, heavy connections to its main source of information, the olfactory bulb. The human medial forebrain bundle remains to be characterized. No one has really discussed the anatomy of the human medial forebrain bundle in any thoroughness. When you lesion this system, you deplete all your psychological resources. This is anhedonia in the extreme, the inability to engage the world and take care of yourself. And it feels probably like profound depression. So here is a great model for depression. And we've actually reversibly lesioned it, and it produces all the classic or depressive phenotypes that people talk about. And the animal is unhappy because it doesn't chirp anymore. It doesn't show happy vocalizations. Can't tickle those animals very well. Okay, here's that same system in cross-section. So this is just the hypothalamic area on one side of the brain, the right side, with third ventricle here, medial lemniscus separating thalamus and hypothalamus. Here's your fornix smack in the middle of it. So what Wallenstein did, remember, he threw the monkey wrench into the field and said, you guys think there's specific motivational systems, hungers, thirsts, and etc. No. It's just a general purpose learning system. And he found this general purpose learning all over the hypothalamus, he said. So I said, no, hold on, Elliot. It's not all over the hypothalamus. Let me take your own data and ask, where are you getting it? 55% of your electrodes are in the dorsolateral quadrant, where you get the best reward, self-stimulation. Dorsomedial is also high. Ventromedial, where we know you have fear and anger systems, it drops off dramatically. And in the ventromedial, you can't find an electrode that does that. So there is a geography to this system. But you can produce eating there in certain animals. You can produce drinking there in certain animals. You can produce gnawing there in certain animals. And what Elliot showed was that these are learned behaviors. They are not intrinsic to the system. Because if you have an eating animal, and you take away the food overnight, but still apply the brain stimulation, in the morning it drinks. Or it gnaws. And if you were a drinking animal, in the morning you're eating or gnawing. And when you throw in the original reward, the animal doesn't go back to it. It sticks with its new behavior. So that's why he said there's full plasticity in the system. And I had arguments with Elliot. He used our data to support his view, which was consistent with his view. But I said, don't say it's just learning and general purpose plasticity. You know, and I know, that there is something fundamental there that's the same in every animal. And we both agree over a beer that, yes, the animal gets excited and moves around and sniffing and, is checking out the world. Every animal shows that, and that never disappears. That is a seeking instinct, a looking for instinct. But he chose not to acknowledge that, for whatever reason. I'm sure there's a deep psychoanalytic reason for that. And, if I have to read someone else's mind, I would probably say it's because of his deeply felt kind of liberal urges that, we all women are created equal. And, we fall into our lot in life and we learn. But, plasticity is the ultimate good. I think it reflected that kind of political philosophy. And I respect it. But down below, we have to respect what evolution gave us. So Wallenstein did show behaviors were completely interchangeable, and I kept saying there's one common feature. It is foraging, seeking. And this full plasticity of the underlying substrates is dramatic and beautiful, but it is not completely plastic. So I think the foraging, expectancy seeking concept still covers as much of the data as any other view, and it is terminology for the primary process level. All other ideas out there in the marketplace are either secondary process concepts, and reward prediction error, Wolfram Schultz at Cambridge, is a tertiary process. So everyone is correct, but they have no way of communicating. So the other thing we found was that every electrode will produce sniffing. Every electrode will produce sniffing. And we decided to throw away our stereotaxic maps of, 2 millimeters this way and this way. We said, we know where it is. Put a hole there, start dropping it down, apply brain stimulation under just enough anesthesia, and when the animal's whiskers start whisking, exploring, we put it there. 100% of those animals were self-stimulators. 100%. And, more recently, 30 years later, we discover that the 50 kilohertz chirp is also a 100% indicator of that process. Wherever you get a stimulus-evoked chirp, chip, 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 you leave your electrode there, that animal will self-stimulate. These are laws of nature at the primary process level. So dopamine, that's the orchestral leader. For very complex neural systems, it does help create an anticipatory state by helping associate stimuli in the world. The higher processes, they're connected. So here's kind of Wolfram Schultz's basic finding. Animals, monkeys are working for food, and initially when the food comes down the chute here, the dopamine cells fire. But as the animal learns that this light, the CS, or sound in this case, predicts the food, the responsivity moves to the predictive stimulus. So it's a reward prediction signal. And if the food reward doesn't come, there's a slight reduction in activity, meaning there's an error. So signaling error. It's wonderful data, but the concept is a tertiary process learning concept. So I'm not going to cover Wolfram's wonderful work, but the consistent tendency of dopamine neurons to fire selectively in response to unexpected rewards. Give them a new treat that's out of the blue, bing, these things start firing like crazy. Okay, so it's not predicting anything, it's just excitement. Wow, I better start sniffing around, maybe there's more of that, which the animal does very intensely and persistently. The tendency of these cells to decrease responding to predictable rewards as they gradually become responsive to stimuli that predict rewarding events. That's basic classical appetitive conditioning. So they do condition, but Jim Olds, in his last 10 years of work, indicated that probably half the cells in the brain show that characteristic. It's not just these cells, it's all over the brain. Everyone wishes to disregard Jim Olds' discovery. So the inhibition that occurs with omission of rewards, this has to lead to engaging the world. Okay? And it does, because if you keep doing stuff like that, the background level of dopamine firing goes up as the animal is starting to search for a new solution. So these Schultz changes reflect information received by the dopamine neurons, but the question is what information or state is being transmitted onward. There's a fundamental undergraduate error. You're measuring from dopamine cells. That means you are looking at the information coming into the dopamine cell, but you are talking about what the dopamine cell is putting forward. It's mixing correlations and causes dramatically. I don't get it. Perhaps an elementary mistake in conceptualizing a neo-behavioristic conception, and the people are alive and love those kinds of conceptions. But I'm not going to criticize that, because but what we can confidently conclude from electrophysiology is that dopamine neurons are indeed listening to a great deal of associative information processed in other parts of the brain. However, it may be unwise to assume that the message passed on by dopamine neurons actually retains the information that the cells have been listening to. It's passing on something new. That's what synapses are for, transformation of information. It is just as likely that instead of a reward prediction error being passed on, dopamine cells actually pass on an urge to behave in a certain characteristic appetitive way, to approach, to explore, to investigate, in a word, to unconditionally seek resources, or if unpredicted rewards emerge out of the sky, to re-energize this urge, because there's bound to be more. If so, the so-called reward prediction error function abstracted from a study of the inputs to the system may tell us little about what dopamine is organizing postsynaptically. So as Walter Freeman emphasized, he's, as you probably know, the son of Freeman, the frontal lobe neurosurgeon. He says each level of neural activation reinterprets the input. Information does not simply get passed on within the brain, but different levels of organization in the brain may speak their own functional languages. So is the functional language here of the mesolimbic? These are the cells he records from. Is it to energize a global psychobehavioral process the way I think it does, or are they passing on prediction errors? I think the mountain of data supports this. Now, Wolfram finds that these lateral A9 cells that cause Parkinson's disease behave just like the cells that in the ventral tegmental area that mediate motivation. That's puzzling. But if they have to listen to the same kind of information, that might make sense. You get the intense reward in this system, which is medial in the brain. And medial means ancient, more ancient. These cells are more lateral in the brain. They do mediate reward, but you gotta push it. And they go into the dorsal striatum, which now everyone is very excited, causes habits, makes your behavior habitual and unconscious. So the ancient system is more conscious than the more recent dopamine system. This causes intense affect. This causes unconscious habit structures. That's a very efficient way for the brain to be built. And we've written piles of theoretical papers on all of this. And look at how much information these cells get. Okay? They get everything you could imagine from down below or close to it. Lots and lots of stuff from up above. So they are listening to what the heck is happening in the world. Then they control this basal ganglia for repetitive learning. And that also gets lots of input. So the system is hyper interconnected, and we know learning is occurring in that system. But what is the primary process? I think seeking is fine. So one of my postdoc students is at NIDA right now. And he's done an interconnectivity map of all the anatomy. Here's how all these systems are interconnected. It's a zoo, basically. And dopamine kind of just, hey, guys, let's play together. It's a conductor of the system. So when he was doing his PhD with me in the mid-90s, we said, let's look at the, kind of nucleus accumbens learning field, and let's put animals in a runway to get sugar water. And they have to run to get it, and they are also allowed to drink as much as they want in a minute. And we found that when you block the dopamine here, the animal stops running. This is getting very slow, centimeters per second, very sluggish. But if you put them in the gold box, they drink just as much sugar. So they still like the sugar, they just don't care to work for it. Just by walking there. It's amazing. So they're not motorically impaired. So this is a wanting-seeking process that's blocked. The liking process is not. And here's what the maze look like. They've got to make a couple turns. Here's a piece of old data that we collected, back in the ancient days, before the Society of Neuroscience, using schedules of reinforcement that Skinner gave us. If you have to chop wood, you keep chopping at a steady rate until you got your wood pile. Then you take a break, have a cigarette, or used to anyway. And which of these schedules of reinforcement is most interesting? It's the fixed interval, because it shows this scallop. You know that you have to do something, but you have to do it every minute, let's say, one time. But the rat is never a philosopher. It's always overeager. So it starts pressing faster and faster and faster and faster and faster and faster, and bang, it gets its reward. Humans work this way, rats work this way, pigeons work this way. Here is the anticipatory curve of the seeking system. So we asked what I think is a profound learning question. Does the seeking system learn spontaneously with no external cues in the world? Okay. So how do we do this? We put an electrode into the system and we just give it the brain stimulation reward on a fixed interval. And what do we look at? Well, the best thing to look at is sniffing, because this system generates sniffing. Or you can look at 50 kilohertz sounds because this system generates those happy sounds. And here's the fixed interval. Every 20 seconds in 4-second blocks. That's all that happens. Initially, the animal just gets excited, sniffing away, exploring like crazy. Usually its breathing rate is about 3 times per second. So it's sniffing 6-7 times a second, and as trials progress, it gets shaped. And with more trials, into a scallop, just like the fixed interval schedule. Okay? You're not training the animal, you're just exposing it to systematic anticipatory arousal, and the system takes on the shape of B.F. Skinner's most famous schedule of reinforcement. Here's chirping that we did more recently, and you get the same scalloping of chirping spontaneously. We are presently trying to bring this into tissue culture. We'd like to have a real learning model, not long-term potentiation that you've all heard about that might be related to learning in some way. We want real anticipatory learning in a tissue slice first, then maybe tissue culture. So we're just setting up the apparatus for that. Don't have any data. We found similar effects with food reward, with social reward, with amphetamine psychostimulant rewards. This is the function of the system, to search, to find, to anticipate. And if you look at the sniffing, when the animal has to press a lever, you can ask the question, does the sniffing, remember he's not a philosopher, he's anticipating eagerly, the first response never produces anything, but it always shows that response. It never becomes a philosopher. 1 second before that first lever press, the animal's nose begins to become engaged. Wow. So this is the mechanism that leads to the lever press. No one's ever followed this up in the past 40 years. But one of my postdocs, Brian Knutson, finally got the message about this seeking system, and when he went to do his next postdoc at NIH, he set up this human brain anticipating monetary rewards. Basically, the first experiments in neuroeconomics. And when the person was playing this kind of gambling game and was about to win big, this system lit up. And now he can predict when a person in a simulated shopping situation, can he predict from the brain images whether the person decides to buy or not to buy? And the answer is quite simple. If this system, this desire system lights up, the person will buy. If the insula lights up, and you know the insula is big in disgust, even little mind, like, oh, I don't really need that, the person will not buy. There is use-dependent plasticity in this system. It sensitizes. Initially, it just says, I just want, I'm eager. And when it sensitizes, the animal says, I want it now, please. Insistence, psychic power. Here's sensitization in our hands. Animals that got psychostimulants twice a day for 10 days. Amphetamine at 5 milligram versus control. The animal shows elevated place preference for that compared to controls. It just seeks food more intensely, even though it's not been trained to change its behavior. This urge goes toward, I want more food. It also goes to, I want more sex. So it just wants everything more. And we give our ADHD kids Ritalin, right? Which is basically possibly sensitizing this system. And at least in our young juvenile rats, this system sensitizes with therapeutic doses of Ritalin. So are we making our kids just more materialistic? Pharmacologically, it's a possibility. is a great model for depression. So we are doing the following depression experiments, saying the positive hedonia of life is diminished because this system becomes unresponsive. We know the separation distress system, as Bowlby taught us, the loss of mother and social support early on is the main source of depression. The lowest part of the system is the most intense emotionally. So we put electrodes in the periaqueductal gray to simulate the separation process. And we are finding evidence that this system becomes rapidly less responsive, as measured by tickling rats. Okay, so this tickling business that produces 50 kilohertz chirps under stimulus control is a good probe for how this system is doing without any further complicated techniques. So we generate a sadness, grief, panic process here in the PAG. Seeking shows reduced arousability. And we also are, after bad experiences, we are giving the animal opportunities to play, or we tickle them to see if it's therapeutic in the downhill cascade. And the answer so far is yes. So this really simple-minded. As the organism is getting depressed, if right after those manipulations you do something positive, you prevent the downhill cascade. You know, I think a 3-year-old would understand that. And with colleagues at University of Bonn, Volker Koenen is the leader of the group, and Thomas Schlapper, they have visualized the human medial forebrain bundle, and they also find the anterior thalamic radiation that maps onto the anatomy of our separation system. Human separation system, as best we can determine from mapping it in other species and human brain imaging like Damasio's 2000 PET paper. So, and they join down here near the ventral tegmental area. So there's a connection between our seeking system and our panic system. And I bet you when a child loses contact with mother, the first thing that happens, you get this massive negative affect from the panic system, but you also activate the seeking system. Because the child, at least little puppy dogs and other animals, they run around like crazy. They run around like, you know, they've lost their heads. Where's mommy? You know, that's a real seeking urge, but it's within a negative affective context. So you can have the seeking urge in the midst of intense negativity. That's why the question mark. This system is not just positive, it is a system that can fit very well with negative experiences. You know, when you are afraid and the predator is far away, you hide, you freeze. When the predator gets certain distance from you, whoosh, there's a flight response. I would bet right now, or hypothesize, that the transition from total immobility to dramatic flight is the system grabbing hold of the seeking system and says, go for it. Seek safety. But that's the hypothesis. No one's pursued that. So here's a closer picture of that. And Volker has described several branches of the medial forebrain bundle, and it's the superior lateral branch that seems to connect up with perhaps the panic grief system. We've also decided to look at the classic history of fine psychosurgery for depression. And after they, stopped the massive lobotomies, they kind of narrowed down the lesions into a kind of ventromedial quadrant where a lot of these systems are concentrated. And they had a surgery called anterior capsulotomy where 48% were improved. Anterior cingulotomy, where a third were improved. Subcaudate tractotomy, again, another third. And if you did limbic leucotomy, which was basically the anterior capsulotomy combined with a subcaudate, you got pretty good effects without all the personality changes that characterized massive lobotomies. So one can ask, you put a seed in the brain and you do diffusion tract imaging. That's so wonderful, probably more wonderful than regular fMRI, to just visualize the connections within the brain. Do these lesions have some convergent connection into something that is meaningful psychologically, the way I just described? And they basically find that the higher kind of what we assume is the separation distress system is connecting up with the ventral tegmental region, even though Helen Mayberg's famous area 25 of the anterior cingulate does not seem to have a direct connection, but there's plenty of possibilities for indirect connections. And here's just sexy images of the diffusion tensor imaging. The blue is the medial forebrain bundle. The orange tan is perhaps a separation distress system. At least the anatomies match up reasonably well, and lead to the hypothesis that the two system separation that Bowlby emphasized and that we mapped in the mammalian brain and the primitive seeking urge are highly interactive systems. And when the negative affect gets too intense, it begins to deplete the resources of the seeking system. And that's what Bowlby would have called the despair response. So from the acute grief response, the diminution of the seeking system, perhaps through dynorphin, anhedonic molecule that fills the medial forebrain bundle, one gets chronic dysphoria of depression. So let's go to the third one. Very quickly, this measure of appetitive desire. Rats love to play. Mice don't. Laboratory mice, but wild mice love it. So it's been bred out of laboratory mice. If you want to do this kind of research in mice, sorry, you're going to be batting your head against the wall. Go to rats. Little mice, laboratory mice don't play. Wild mice play just fine. And we discovered that the animals are making these chirpy sounds, going wild. And we studied play vocalization for a long time. Remember, this playful circuitry is generating joyful feelings because we know this sound is rewarding. The rat in general has two affective vocalization, the chirpy ones, for all good things in the world, and ugly, life is bad sounds for practically everything that is bad in the world. They got great positive, negative affect measures that have circuits in the brain to be analyzed. You can see there are two different types here. One's a longer type, one's a short type. And when you stretch it out, there is a short one that is monotonous, flat, that is simply not full of joy. It's like maybe nervous laughter. But the longer one has this wonderful frequency modulated tail, and that is the real Duchenne laughter, sincere laughter. That is what predicts reward. This one does not predict reward when you're mapping. And when you stretch out this thing, you just get when things are really nervous, but not when they're really bad. So when it's intense fear, the animal shuts up completely. It's only in anticipation that things are going to be bad. So it's a nice, subtle psychological measure. So we bred for these by essentially getting a random line and selecting animals that chirped a lot to a standard stimulus, which was tickling. So we tickle rats, and they love it. when they're young, and they become bonded to you. And those that didn't chirp a lot. So eventually, we're on 20th generation. The sad animals are dying out, by the way. They're not reproducing. So there's some problem. And it's the second time in breeding we've seen that. So what is happening in the sad system, we don't know. But we were just selecting for 50s. And you have this happy line and this sad line. And spontaneously, the other side went in reverse. The sad line started showing more of these complaints. And the happy line, fewer complaints. So this was not selected for, but indicates that the genetics has some kind of interactive balance in the system. And here's a tickling paradigm. It's very rapid. No tickle, no chirps to lots of chirps. It's incredibly robust. And then when Jeff Bergdorf mapped this for his dissertation, here's what he got. It ran along the mesolimbic dopamine system. And wherever he got it, it was rewarding. So summary, these 50 kilohertz chirps indicate positive affective states. Euphoric drugs and brain stimulation elicit them. Euphoria in humans and 50 kilohertz chirps in animals are modulated by dopamine. Here's the dopamine story. Looking at these monotonous ones during tickling and rewarding brain stimulation. So here's the monotonous flat call, and here are the happy chirping calls. And when you block dopamine, you do not block the 20s, you do not block the 50 flat ones, but the frequency modulated are dramatically reduced. Same goes for rewarding brain stimulation. No effect on 20s or the flat ones, but the frequency modulated 50s are dramatically reduced. So they're just proof of principle that everything is working the way it is supposed to be working. So now we have an external indicator that's been validated where the animal can tell us where it is in affective space. Imagine how valuable that is. Prior to that, it was just behavior. Now we have a psychological state that the animal spontaneously informs us about. Not intending to inform, but it's very informative. If you lesion the area, remember you get a depressive state. The 50 kilohertz go down. So here is animals 1 week after the lesion. They're the same before. And these are small lesions. So the animal's not really dramatically debilitated. And it's not a matter that you can't vocalize. The animal can vocalize because the negative ones are going up. So again, you see this balance of good and bad happening spontaneously. They can be used to measure drug reward. So if the animal's gotten good drugs in a situation and then you put it back, it chirps like crazy. It shows very few of these bad calls. On the other hand, the ugly calls, if you give them a mild dysphoric, block the opioid system, there's more of these negative calls. If you give them a bellyache with lithium chloride, you get even more of these negative calls. So that's a general purpose type of thing. You can put your amphetamines directly into the learning machinery, the nucleus accumbens, and as you increase the dose, the habit structure part does not generate the vocalizations. That's where dorsal striatum, just habitual behavior. The more kind of general purpose dopamine part does increase it markedly as a function of dose, but if you put it into the most rewarding parts, you can use the psychostimulants as a direct driver of the positive calls. And in our lines, if you give a happy line amphetamine here, they really go super chirpy. And the sad animals are kind of unresponsive to the amphetamine. So the whole puzzle box fits together beautifully. But moving on, social processes. Seeking is big in maternal behavior. The maternal system goes right smack into the ventral tegmental area and drives all the activities a mother has to do in order to take care of the babies. Building nests, if the babies have gotten lost, carrying them back to the nest, retrieving. And these bonding chemistries, we found opioids were a big part of the bond. Oxytocin, lots of people are finding, including perhaps humans. But here it's not, oxytocin does not produce a positive affect. That's one bit that's missing from the discourse. No one can show, and we tried over and over again, that animals like to go back to a place where you put oxytocin in their brain. They will go back if there's other animals there, so there's already a social reward. The oxytocin will increase the social reward. But by itself, it will not produce social reward. But it does produce some kind of a psychological response. And people are calling it everything from love to trust to, just feeling squeaky good about life. There's no evidence for that. But it's doing something psychologically, and we don't have the right word. My best bet right now is it dramatically increases confidence. And guess what? If you have confidence, you will trust other people more. Unless you have a borderline personality disorder, and Bartz's work at Mount Sinai indicates that those people getting oxytocin up the nose become less trusting. Okay? Because they're insecure, and now they can express their insecurity like, screw you. I'm feeling confident, and I think you're taking advantage of me. So confidence can easily explain the paradoxes and all the neuroeconomic experiments that are being done, and no one has yet done the simple measure of confidence after oxytocin. But it explains our socially facilitated reward, because of course you put an animal in with another animal, and the animal is happy, but it's kind of insecure. What's going to happen? There's a new world. A lot of stuff can happen. You give oxytocin and feel secure, it's a much more positive situation. We tested this in little chickens 20 years ago, and we put little chickens that are very insecure by themselves, so when you put them in a large room like this, they move like a flock, tight-knit. And when they are given oxytocin, they still move like a flock, but they spread out farther. That, to me, was a test of confidence. We put it into the quail brain. Oxytocin makes you peaceful, less aggressive, and male quail are super aggressive. First time they get together, they bang each other's heads. And the guy that bangs more and the other guy gets into a squat, that remains their typical interrelationship. When we gave one of the animals oxytocin, it got its head pecked off because it was peaceful. When that animal wouldn't have oxytocin, it would always be the bottom bird. But when we took the oxytocin away the next day, the animal came and pounded the other animal into submission. It probably indicated you were peaceful, and you said, well, it didn't affect me a lot. I just wasn't aggressive, and I took it, but I remember that you were a prick. Anyway, that's our interpretation. It'll be in the title. You are a prick. The security, what a mother really needs is confidence, a can-do attitude, and that is what young mothers report who have good maternal stuff together. I can do it all of a sudden. That security that has to be passed down to the baby. I wish some human studies would measure that. One of my PhD students from University of Rome is wanting to do that study with one of the great European oxytocin people. So, Michael Meany, all these great epigenetic effects. We had Steve Sumi a while ago. You know, they got the 24/7 methylation machine, Moishe Schiff, where, you know, every gene can be looked at their methylation patterns. It keeps cranking it out. Knowledge, you know, by the pound, without any effort. And what Steve found was, and with Michael Meany and Moishe Schiff, that in the rhesus monkeys that are brought up in two ways, those that are brought up with loving mom and are well regulated, compared to the ones that are brought up with peers, namely, they are brought up in a gang of toughsters, and they become very kind of, you know, gotta fight, gotta stay ahead of the gang. 1/3 of the genome was differentially methylated by those two rearing patterns. That's more than you can think about. Here's something you can wrap your mind around. These ancient emotional forces are things we share with the other mammals. And until the scientists begin to recognize what the data is saying loud and clear for the last 40 plus years, we're not going to make progress that connects up with psychiatric issues as much as we could. If we accept the animals and these emotional powers and try to understand them as fully as modern neuroscience can, we will make much more rapid progress. The problem is the lack of conversation. When intelligent humans stop talking to each other in reasonable ways, you get the madness of modern science, where technology takes us to ruthless reductionism and the nature of the organism is lost in the muddle. Oh, I better control myself. Opioid reward is an attachment mechanism. It's coming back gangbusters now. It was lost through most of the late 80s and most of the 90s, but now it's coming back. Thank goodness. It's such a rich and affective system that is much underutilized in modern psychiatry. That's the long story. It could be used. We have chosen not to use it with skill and understanding. We are not even taking care of our children as well as we could. Spock said it after John Watson. Just leave the kids be by themselves in a dark room when they go to sleep. That is the way modern humans rear modern humans. And, you know, no other primate mother puts their child on an empty limb hanging there all night. You know, this issue that maternal bonding insists that mother and child sleep together for a while, it would dramatically change, I think, the affective face of the earth. But we are in a spin. And tickling, you know, we can't touch our kids anymore because there are, you know, assholes out there that do stupid things. There probably wouldn't be if they had been able to sleep with their moms. So we have these self-fulfilling unprophecies. Oh, what a spin we're in. So what are we waiting for? I had 5 French documentary filmmakers doing a special on laughter, and they thought the laughter should be given attention, and they gave me a little video clip unedited of, you know, God, you get 5 guys that are smoking and chatting, and they want to take pictures of rats playing, and so you spend a lot of time with your rats that, you know, you are the secure base. It's okay that these strange and weird guys are going to be filling the room with strange French smells and nicotine all over their fingers. And I played their game, and the rats were dramatically less playful. They were very curious of the ambiance, the new ambiance. But let me at least show the video that they shared with me a couple days ago. The audio on this is very bad. You will not hear the rats chirping because it's a separate file that needs to be filtered and then added on to the video. But if you haven't seen the rats chirping, just Google laughing rats, and the first thing you will get is someone having put me on the web from 1998 when the first video was made by BBC. Let's move me along here. I'm lost in the other room. Here that have not played with each other before, and we hope they will play. Now, I had only tickled these animals, so they'd already seen the video of tickling, which went reasonably well. Now, all of a sudden, they're together for the first time for many, many days because they have to get bonded to me in order to show the phenomena. If they come out of their family, they could give a crap that I'm tickling them. They say, what is this about? Are not going to be very courageous. They gather together. Inside, I'm saying, come on, guys, please play. Maybe if we're lucky, they will start to play because they've been living alone. Oh my, they're not going to do anything. I'm going to just sit here. But these have been tickled here. Yeah. I was going to tickle them after they started playing, but I might have to tickle them before they start playing. Get them all excited. So I figured I would have to jazz them up. Here I go. I'm going, well... Oh, something's happening. You know. Get lively. Oh, half a minute, all of a sudden, start playing. Kind of energy. Well, there's a play movement. Yeah. Oh, looks like they want to play. That's amazing. They're listening to me too. Come on, guys. You're probably smelling that big old rat that was in here before. Yeah, there was a big one there before, huh? If it would have been a male, it would have been very scary, but it was a female. Because you can still tickle old females, you can't tickle old males. Okay, except in the human species with some... Well, it's starting. Verbal tickle. Mother Nature and its impulses are taking over. And they kind of pounce on each other's back. That's an indication they want to play. And then they turn around and they wrestle a little bit. But these guys are still a little timid. They're getting used to it. They're probably wondering where I am, since I'm the only one that's been tickling them. Now they have to tickle each other. Oh, they do that pretty well. Doesn't require much learning. That's because it's instinctual. It's built into their nervous system. But I think I need to stir them up. Took one look at those French guys and he said, what's that about? So let's go, guys. Here we go. Whoa. So now I'm jazzing them up with a little tickle. Come on. Come on. Come on. Come on. Where's that tickle? Yes, I'm the guy that's been tickling you. Okay. There you go. Now they're much more flexible already. So they're more confident. We know it releases brain opioids. They know I'm around, that if they need a tickle, I will come save them. Pretty good data that releases dopamine. Right now we're trying to see whether the bursting of dopamine cells is reflected in the chirp itself. Don't have data on that, but so they're chirping all the time. Chirp, 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 especially when they're jumping and chasing. Oh, my goodness, I said the right word. And it's really a wrestling match. I had a famous psychologist, Robert Plutchik, visit the lab, 10 years ago, 20 years ago. And I said Rob was a great evolutionist, and he hadn't ever talked about play. And I said, Rob, I'm going to show you something in the laboratory. And I brought him up to the lab and just put a couple of animals together under red lights where they're very comfortable. And they started wrestling with each other. And he was just amazed. He looked at it and said, Jaak, how did you train the animals to fight that way? And I said, Rob, I didn't train them. Evolution did. And by the way, they're not fighting, they're playing. I said, playing? I have to learn about that.